So now these two mag and these acyl groups are now we will use these two acyl groups for beta oxidation, and from that we will get ATP. Now listen, for beta oxidation we need to transfer these acyl groups, from here to here. That means the enzymes for beta oxidation of fatty acids are present in the inner mitochondrial matrix. That means this acyl group will have to cross two barriers. Why I am calling this as two barriers? Because these groups are polar, and this membrane is identical with cell membrane. Why? Because according to Robertson unit membrane concepts, all the living body membranes are made up of same type of thing, that is phospholipid bilayer. Okay, that means it will have to pass through two barriers, that is mitochondrial outer membrane, mitochondrial inner membrane, then into the matrix. Okay, now listen, there are two mechanisms by which these two acyl groups are converted to some other chemical compounds, and they are then transferred to the mitochondrial matrix. Now listen, what are the basic mechanisms? You should now understand. Suppose I am rubbing this cell for a minute so that we can talk in details about what the reactions are happening, which will actually lead to the entrance of acyl groups. Now listen, suppose this is a mitochondria and this is inner mitochondrial membrane. Now listen with all your attention. Now you should always understand that suppose this is a cell. Now we know that here there is fatty acids. Now where from we are getting this fatty acid? Now listen, acyl groups are what? Acyl groups are like this. Acyl groups are like this. These are acyl groups. That means they have a carboxyl end and an arch in here. Now listen, there are many H+, which are moving here and there in the cell itself. Now listen, when this H+ will now react with this particular oxygen, then what will happen? It will be converted into an fatty acid. Okay, now the question is this: how will this fatty acid enter the mitochondrial matrix? Okay, now listen with all your attention. There are many sources which say actually that these fatty acids actually react with a special protein known by the name of albumin. Okay, now listen, there is a basic difference of understanding here. Listen, there are two schools which believe completely different things. Now listen, first things you need to understand is, one school believes that free fatty acids which are present in the cell, in the blood, they bind with albumin. They bind with albumin, and these two as a whole enters. Okay, and these two as a whole enters. And other school of thought says that fatty acids which are present inside the cell, they bind with albumin, and then they react with some proteins or something else to enter the mitochondrial matrix. Now listen, both the thoughts are correct. Why? Because there is a strong evidence that free fatty acids obviously reacts with albumin. Okay, why? Because free fatty acids are in most of the cases they are not much polar. They have only one negative charge. That means they need some additional protein carrier so that they can become sufficiently polar to move around blood, which is made up of mainly plasma, and the most important constituent of plasma is water, which is polar. Now we are not going into the details of these things. Okay, now our main aim is how we can transfer this fatty acid into mitochondrial matrix. This should be our first and the most important aim. Okay, this is our mitochondrial matrix. Okay, now let's talk about this. Listen, whenever, suppose, listen, suppose this is a fatty acid. Now listen, suppose this is a fatty acid. Now first things first you need to understand is, whenever you want to take fatty acid from here, from cytoplasm to mitochondrial matrix, first thing you will have to do is you will have to charge your fatty acid. You will have to charge your fatty acid. What do you mean by charging of fatty acid? That means you will have to add S-CoA, that is coenzyme A to this fatty acid. That means it will become fatty acyl CoA. Okay, first thing you will have to understand is this, that before you can transfer this fatty acid into the mitochondrial matrix, you will have to charge. The first step therefore is known as charging of fatty acid. Charging of fatty acid. Now this charging of fatty acid is actually done on the surface of outer membrane of mitochondria. That means there is an enzyme which is actually performing this thing, that is it is converting fatty acids into fatty acyl CoA. Okay, now what is the name of this enzyme? It is known by the name of acyl CoA synthase. It is known by the name of acyl CoA synthase. Okay, it is known by the name of acyl CoA synthase. Okay, clear? So this is the first step. So now you want to take this fatty acid into mitochondrial matrix. Now for this, the first step is charging of fatty acid. The product is fatty acyl CoA and it is done by acyl CoA synthase, an enzyme which is present on the mitochondrial outer surface, which is attached with mitochondrial outer surface. Clear? Okay. Now listen. The second part you need to understand is listen. The second part you need to understand is now this fatty acyl CoA looks like this, okay? Now this fatty acyl CoA can now enter, now it is permeable only to this first layer or first membrane of mitochondria, that is it can now easily enter first layer. Now listen, there is another barrier, okay? Listen, it has successfully reached, this fatty acyl CoA has successfully reached the intermembrane space of mitochondria. This is known by the name of intermembrane space. Intermembrane space. Now you need to understand that now fatty acyl CoA has successfully reached the intermembrane space, right? Now intermembrane space again is a very polar area where H plus is there more, or in some areas there is H plus less, so that's why it cannot stay there for a long time. That means it will have to now enter, thus it will have to now penetrate the second mitochondrial membrane. It will now have to enter the second mitochondrial membrane fast. Now to do that, what it will have to do is it will have to take the help of an enzyme, and this enzyme, what will it do is actually, this enzyme is actually present or is attached this enzyme is actually attached on the inner side of the outer membrane of mitochondria. That is a basic difference between acyl CoA synthase and this enzyme. That means both are present on the outer surface, but one is projected towards the outside and one is projected towards the inside, that is intermembrane space. Now what will this enzyme do is, this enzyme will take up this fatty acyl CoA and it will add another thing to it known by the name of carnitine. It is known by the name of carnitine. Okay. What is carnitine? Carnitine is actually made up of lysine and methionine. Okay, it's an alcohol. Okay. Now you should understand what carnitine is doing. Okay, listen. Carnitine is like a ticket using which it can enter this surface. That means carnitine is the ticket just to overcome the barrier of inner mitochondrial membrane. Now what is the importance of this enzyme? Now listen, this enzyme has two sides. In the first side it will take this fatty acyl CoA, in the second side it will take carnitine and it will take out this S-CoA. It will kick out this S-CoA. Okay, and instead of this S-CoA, it will add carnitine. That means what is now entering is fatty acyl carnitine, right? What is now entering the mitochondrial matrix is fatty acyl carnitine. That means RCO carnitine. Okay. Now listen. So fatty acid or fatty acid has now successfully reached where? The fatty acid has now successfully reached the mitochondrial matrix. But the problem is this, you need to understand, that is the problem with your, most of the times, with service, that is you wanted service for money, but after getting the service, you know that you don't have the time to enjoy the money. Listen, that's the problem with this. It was fatty acid was very much excited to get into mitochondrial matrix, but when it came to mitochondrial matrix, it is no longer fatty acid, it is fatty acyl carnitine. That's the problem. That means life gives this fatty acyl carnitine a second chance so that it can again become fatty acid. What is that? There is an enzyme which is present in the inner mitochondrial membrane and projected towards the mitochondrial matrix. This will take this fatty acyl carnitine, it will take away the carnitine, okay, it will take away the carnitine and attached to this fatty acyl carnitine. Now listen, this enzyme will take away the carnitine and to this fatty acyl carnitine it will add S-CoA. Okay. So what is the name of these two enzymes, right? Okay. So now what is the name of this enzyme and this enzyme? Listen, both have same name, that is carnitine palmitoyl transferase. Now listen, in short it is known by the name of CPT. Now listen, this one is known by the name of CPT1 and this is known by the name of CPT2. Okay, this is known by the name of CPT1 and CPT2. Now listen, now what we have got is actually we have successfully carried this fatty acid into mitochondrial matrix, but now also it is not only fatty acid, it is fatty acyl CoA. Okay, now we will convert this fatty acid into acyl CoA and acetyl CoA, and in this process we will get some ATP via ATS. Okay, now let's start. Listen, now listen, at some point of time you will have to obey someone, right? Like you may have to obey your parents, you are now obeying your teachers. Okay, now listen, in beta oxidation you will have to obey someone, and this is actually a mnemonic using which you can actually understand what are the steps of beta oxidation. Listen, beta oxidation steps are simple. These are biomolecular or biochemical steps which are divided into 4 categories. Okay, they are oxidation, hydration, again oxidation, and then thiolase. Okay, now listen, now that means the acyl group will first undergo oxidation, then hydration, then again oxidation, then thiolytic reaction. Now listen with all your attention. How to remember this step by step thing? Listen. You will have to remember this by obey him on this. That's it. Obey him on this. That's a very basic, thing you need to understand before you can proceed for beta oxidation. Listen. Now, suppose this is your fatty acyl CoA. Okay, this is your fatty acyl CoA. Now listen with all your attention possible. Now, this will be converted into acetyl CoA and acyl CoA having 2 carbon atom less. Suppose this acyl CoA has 16 carbon, then at the end of fatty acid, beta oxidation of fatty acid, you will get is acetyl CoA and an acyl CoA having 14 carbons. That means in beta oxidation is a continuous process. Okay, that means you start with a large number of carbons, then at each circle you will lose 2 carbons, and these 2 carbons are actually converted into acetyl CoA, and these acetyl CoA can now enter into Krebs cycle and give you ATP. And these acyl CoA will again join the beta oxidation pathway. Okay. Now listen. This is carbon number 1. This is carbon number 2. This is carbon number 3. This is carbon number 4. Suppose. You know what is the beta carbon? This is the beta carbon. That means the beta oxidation pathway will go through these, will actually involve only these 2 carbons, 2 and 3. That's it. Okay. Okay, let's start. Now listen. Now we will have to first oxidize this chain. This is the first step. Oxidize this chain. Now listen. You need to understand that how we can oxidize this fatty acyl CoA. Listen. Before that, you need to understand what is oxidation. Go back to molecular biology videos, go back to organic chemistry videos, you will understand what is oxidation. There are basically 3 definitions of oxidation. That is removal of hydrogen, addition of oxygen, and removal of electrons. Right? Now listen. What we will have to do is here, we will have to remove hydrogen. Okay? Now listen. From where we will have to remove hydrogen? From carbon number 2 and 3. Listen. All the reactions which are taking place will involve only carbon number 2 and 3. Okay? That means during exam you will have to draw at least 4 carbons. When you are drawing a general beta oxidation pathway, you draw 4 carbons first. In the first carbon, you will have to attach the carboxyl group together with S CoA. This is nothing but a carboxyl group. Listen, it's nothing but this. And if you remove this negative, then add S CoA in biochemistry. Okay? Now listen. So, the first step is the oxidation, and what is the result? The result is this. It's so simple. Okay, the result is this. Now, before I get back to the details of this result, we should know two things. That is, where are these two additional hydrogen gone? That means there were two additional hydrogen here. That means there was one hydrogen here and here, right? So what we have done is we have removed this hydrogen and this hydrogen, right? We have removed this hydrogen and this hydrogen, right? That means we are removing hydrogens. That means the enzymes, listen, in biochemistry, with each and every arrow, remember these two things. That is, what is the enzyme used whenever in biochemistry? Suppose I am writing A to B. In biochemistry, whenever I am using an arrow, listen, you will have to understand these two basic things. That means whenever in biochemistry A is converted into B, that means something is going out or something has been added. That means in the downside of the arrow, in the downside of the arrow, you will have to write the thing which is coming out or which is going in, and upside will contain the name of that enzyme which is helping you to do this step. That means here I can use left or right. Here what I will have to do is I will have to give, or I will have to provide you some necessary information using which we can understand that which particular thing has taken this hydrogen out, right? Listen, what is the work of an enzyme? The work of an enzyme is it will break these bonds, right? That's it. The work of an enzyme is to break these bonds. Now listen with all your attention. The work of an enzyme is to break these bonds. Now what will happen is you need to understand, that is, these two hydrogens are now free. Now whenever any enzyme has taken this place, has applied its power and cut these two hydrogen or any hydrogen out of a biochemical substance, then these hydrogens are taken up by some hydrogen carrier or electron carrier known by the name of FAD or NAD. Now listen with all your attention. Something called sigma and pi bonds. These are what? Sigma bonds, right? That's true. But you need to understand one thing. These are low energy sigma bonds. Why? Listen. You know what is high energy sigma bond? Suppose this is a polar covalent bond. Okay. Now listen. If I apply enzyme and if I apply energy, and this becomes this, that means H dot and Cl dot. What is this? Free radical. If from this scenario I will have to generate free radicals, then we need sufficient energy, and this will be taken, and this sufficient energy will be provided by some enzyme, suppose E1, and this E1 will give only this hydrogen to some very good reducing group, one of the best reducing group of NAD. But listen, do you think this enzyme, suppose there is an enzyme, I will write the enzyme name of this enzyme, suppose this is an enzyme E1. Do you think this enzyme is doing enough work to cut these bonds or to destroy these bonds? No, because these bonds are non-polar bonds. These are non-polar bonds. Okay, that's the very basic thing you need to understand. That means these bonds can be easily broken. Listen, that means this enzyme is doing not much work. That means why I am telling these things, because the hydrogen which are coming out are not much powerful. That means they don't need strong reducing agent like NAD. These hydrogen can also be taken up by those reducing agents which have low delta G or Gibbs free energy. Listen, what is the basic difference? When I ask these things, people just look at my eyes. What is the basic difference between NAD and FAD? NAD accepts hydrogen which have greater delta G. Okay, lower delta G hydrogens are taken up by FAD. Now listen, these hydrogens have lower delta G. That means it will be taken up by FAD, and then they will be converted into FADH2. Okay. Now, so with each arrow, we will discuss two things. That is, what is the cofactor or something like that, and what is the name of the enzyme. Listen, before I write the exact name of this enzyme, you can very well guess the name of the enzyme. Why? Because it will be dehydrogenase. It will be dehydrogenase. It is taking out the hydrogen, that's why it will be known by the name of dehydrogenase. What is the exact name of this dehydrogenase? Listen, if you want to give name of any enzyme, just understand the name of this particular product, understand the name of the substrate on which this dehydrogenase is acting, that is acyl or fatty acyl CoA dehydrogenase, which is simply known by the name of acyl CoA dehydrogenase. Acyl CoA dehydrogenase. Okay, so we are done with step 1, that is oxidation. Okay, we are done with step 1. Okay,